For unicellular organisms, cell division or fission leads to the creation of new individuals. Many different patterns of fissions have been observed. Many bacteria and protozoa simply split into two equal halves during cell division. In organisms such as amoeba, the splitting of the two cells during divisions can take place in any plane. Some unicellular organisms show some organization in their bodies, such as in leishmania, which have empty structure at only under the cell. Intervention occurs in definite orientation in relation to the structures. Other single-celled organisms such as the malarial parasite Plasmodium divide into many daughter cells simultaneously by multiple fission. In multicellular organisms with relatively simple body organization, simple reproductive methods can still work. Spirogyra, for example, simply breaks up into smaller pieces upon maturation. These pieces or fragments grow into new individuals. This is not true for all multicellular organisms. They cannot simply divide cell by cell. The reason is that many multicellular organisms, as I have seen, are not simply a random collection of cells. Specialized specialized cells are organized as tissues, and tissues are organized into organs, which have to be placed at the definite positions in the body. In such a carefully organized situation, cell by cell division would be impractical. Multicellular organizations therefore need to use more complex ways of reproduction. A basic strategy used in multicellular organisms that for different cell types perform different specialized functions. For reproduction achieved from a single cell type. So the single cell type in the organism is capable of growing, multiplying and making other cell types under the right circumstances. Many fully differentiated organisms have the ability to rise to new organisms in their body parts. If the individual is somehow cut and broken into many pieces, many of these pieces go into separate individuals like Hydra and Planaria, cut into many pieces, and each piece grows into a complete organism. This is known as regeneration. Regeneration is carried out by specialized cells. These cells proliferate and make large number of cells. From this mass of cells, different cells undergo changes to become various cell types and tissues. These changes take place in an organized sequence referred to as development. However, regeneration is not the same as reproduction, since most organisms would not normally depend on being cut up to be able to reproduce. Organisms such as Hydra use regenerative cells for reproduction in the process of budding. In Hydra, a bud develops as an outgrowth due to repeated cell division at one end. These buds develop into individuals, and when detached from the parent body, grow into independent individuals. Vegetative propagation. There are many plants in which parts like root, stem, and leaves develop into new plants under appropriate conditions. Unlike in most animals, plants need to use such a made for reproduction. This property of vegetative repropagation is used in methods such as layering or grafting, or grow into many plants like sugarcane, roses, or grapes for agricultural purposes. Plants raised by vegetative propagation can bear flowers and fruits earlier than those produced from seeds. Such methods may also make possible the propagation of plants such as banana, orange, rose, and jasmine that they have lost the capacity to produce seeds. Another advantages of vegetative propagation is that all plants produced are genetically similar enough to the parent plant to have all its characteristics. Tissue culture. In tissue culture, new plants are grown by removing tissue or separating the growing tip of plant. The cells are then placed in artificial medium, divide rapidly to form a group of cells or callus. The callus is transferred to another medium containing hormones for growth and differentiation. The plantlets are then placed in the soil so that they can grow into mature plants. Using tissue culture, many plants can be grown from one plant in disease-free conditions. This technique is commonly used for ornamental plants. Even in many multicellular organisms, specific reproductive parts can be identified. Thread-like structure can be developed on the Bread mold rhizospores, they are not reproductive parts. On the other hand, tiny blob on a stick structures are involved in reproduction. The blobs are sporangia, which contain cells or spores that can eventually develop into rhizospores individuals. The spores are covered by thick walls that protect them until they grow into contact on a moist surface.